the guy in the field. So if you see me out in the field, please say hello. And if I'm on a boat, you can honk, say hi. It's all good. But I also wanted to apologize to you guys because I have a lot of notes and things. I have a lot that I have to get through. These other guys, man, I was like, how do you do all that so quickly? So if I didn't have notes with me, I'd be talking about giraffes within 5 minutes. So we're going to hopefully stay on topic. But I wanted to start this with what the Jefferson Project is. It's a collaboration between RPI, IBM, and the Lake George Association. And we all have our different strengths. You know, we have this robust science and advanced computer models and community engagement. And the point of all that is that we can work together and use those strengths to accomplish our mission. So there's a picture there of the sensor network that I'll get back to. And I also want to talk about the sensor network because we have 3 major platforms that you probably see around the lake, and it's fun to talk about them because they're very visible to you guys. So the vertical profilers are something that are really, really important to us. They're capable of monitoring basically everything on the water, below the water, and above the water. So there's a weather station there up on top. We have pyronometers to measure light. It's all run by solar. You can see the panel there. And inside of that white, we call it a doghouse. But in the doghouse, there's a winch that sends a sensor up and down the water column. You know, Jim was talking about the YSI sensor. We use the same platforms, same sensors, and it goes all the way up and down the water column, and it's able to capture data at every single step, all the way down and all the way back up. Really powerful stuff. We also have weather stations. We have 7 different weather stations around the lake, and that's really important because so many of the things I'm going to talk about today are so weather dependent. So all the things happening around this lake, it all matters. It's the weather. Every previous speaker before me, it's all weather dependent. And we also have we have 11 tributary stations. So these are probably a little bit more hidden. We try to put them out of the public eye because people like to mess with things. But these are extremely important because, once again, all these presenters were talking about different things involving streams and runoff and all these nutrients and salt. So without these stream stations, we really wouldn't have a good understanding of what's going into the lake, and that's a huge part of it. So on the picture over there, you can see that there is a very vast sensor network, and you can go on the jeffersonproject.live website to see parts of this, even with some live data. And I wanted to talk about the 3 different vertical profilers and where we put them and what we're looking at with each of those, because people always ask me, why do the vertical profilers move? It's because we always have different research questions. So for 2024, we have one in Northwest Bay, and we're looking at stream intrusion. And I will talk about all these things in more detail later in the slides. But stream intrusion, again, really important to everything we brought up so far. Harris Bay, we're looking at harmful algal blooms. Harris Bay, for some reason, has been one of the hotbeds for harmful algal blooms. So we've pretty much always had one there trying to figure that out. And then we have one in Caldwell Basin, which is not a name that people usually recognize very well. So it's the lower star there on the map. So there we're looking at a deep water oxygen depletion, which is really just a fancy way of saying it's running out of oxygen at the bottom of the lake. So we'll talk about that as well. And the big fancy plot that you see there is one of the profile plots that we can get from the vertical profiler. So this is taken in 2023 from October 5th through October 21st. And there are thousands and thousands of data points in every single vertical line, and that's how you make these plots. And those separations in color are going to tell you the amount of oxygen in the water. So we'll get into that. So for the overview, we're going to talk about what are harmful algal blooms. Really, really big. Matt already knows the deal. So really, really big topic here, and I know it's something we talk about a lot at Lake George. So there's a lot to get through. So again, I'm going to try to stay on my points. But has anyone here actually seen a harmful algal bloom? Nice. Okay, great. Yeah, there's at least some. So, and I want to make some caveats throughout this. There will be notations of this is not Lake George because I'm not trying to cause any panic. There are other places that have really extreme cyanobacteria events that I have personally experienced. So it was a very eye-opening experience for me. But even in the middle there, we're using microscopes to look at these things down at the microscopic level to try to understand what's going on. So let's just talk about what are cyanobacteria. They are also known as blue-green algae, and they are actually bacteria. So we'll get into that as well. It's really confusing because some of the terminology can say algae, bacteria, but we'll talk about it. And they are one of the oldest organisms on the planet. So they're responsible for the production of oxygen through photosynthesis. They were the first organism on the planet to photosynthesize. So as you sit here breathing, you can thank cyanobacteria. They're not meant to be the enemy. They just have a little bit of problems. And they're also primary producers. That means that they are the base of the food web. So everything in Lake George counts on cyanobacteria and other parts of the basal layer of the food web, or else they wouldn't be able to live the way that they do. So we really actually count on them as well. And here's the fun part. Cyanobacteria are not true algae. They are prokaryotic versus eukaryotic. And I know I'm giving you flashbacks to high school biology, but there will there will not be a test, I promise. But just for fun, does anybody remember what that means? What does prokaryotic mean? Nucleus. There we go. Look at you guys. Yeah, so prokaryotic means that they don't have a nucleus and organelles that are contained in an organized way. Just means everything's kind of free-floating inside. Eukaryotic means that they do have a nucleus and organelles. So that means that cyanobacteria and algae are in completely different domains. They may function similarly, but they're very, very different. And with true algae, they have different photosynthetic pigments. So you have blue-green with cyanobacteria, but with true algae, you have greens, browns, and even reds. So when you're looking at things that are growing on your rocks, those are algae. Those can be cyanobacteria. They come in many, many different forms. And it's really important to talk about their reproduction as well. So various strategies. Cyanobacteria are almost exclusively asexual. It means that they don't have sexual reproduction, and because of that, they're mostly cloning themselves. True algae, they do have some asexual reproduction, but they also have some sexual reproduction, which means that whenever they reproduce, there's actually a recombination of their genes. So what that's telling you is that with cyanobacteria, they are able to reproduce quickly, and they're mostly cloning, and so their genes are not changing as they're growing in population. They're almost all clones of one another. With algae, they can evolve in a more unique way. And as I said, algae are natural and important. They're the base of the food web and really important for oxygen production, not just for what we breathe, but everything in the lake as well. Let's see, make sure I got everything. Yeah, I think we're pretty good. So I want to talk about all about blooms and the different characteristics. And whenever you see things in the lake, I get calls all the time from people and saying, I saw a bloom, I saw this, I saw that. So they're not always cyanobacteria. There are many other things that do bloom. It's really common that green algae blooms. There's filamentous algal blooms, or FABs, all over the place. That's one of my favorite acronyms. And there are many different types of cyanobacteria that can cause blooms. So, and again, this picture was not taken at Lake George, but that one was. And you can thank the waterkeeper for that. He put me onto this one in Diamond Point, and I think we determined that there was probably an issue with their septic system, something leaking into the lake at one small point, and this filamentous algae just grew out of control so quickly. When I was an undergraduate at the University of Pittsburgh, we worked with culture pools and tadpoles and did all sorts of experiments, and I had to sit there for over an hour every single day taking filamentous algae out of culture pools. I can tell you it was extremely fun, and I never got sick of it. But it was just incredible to me how quickly it could grow. So sometimes people get so surprised because they walk on their dock and they're like, well, that wasn't there yesterday. Why is this happening? So not usually a cause for concern, but you do want to inspect maybe why that's happening. And with this other photo here, I wanted to highlight how many different kinds of cyanobacteria can cause blooms, because they all come with their own different characteristics and different life history traits. So it's important to know that it isn't just one organism. So make sure to skip. I think it'd be really important to include things in here that what are HABs and what are not HABs, because again, people get a little bit up in arms sometimes whenever they see the wrong thing, and they, they're doing the right thing. They call us, they call the waterkeeper, they call the LGA, and they say, hey, I saw something. So this is more meant to educate you on the different possibilities and what HABs are and how they're different. So in this first one here, we have filamentous algae. Again, it's going to be a topic. It makes these long threads. I mean, you can poke a stick in it and actually pick it up, as you can see there, versus this more like spilled kind of paint. It's honestly kind of beautiful sometimes. It's unfortunate because it's so pretty, but yet it can be so bad. And this spilled paint on the surface of the water, it's actually millions and millions of colonies of cyanobacteria. They do form into these colonies, so if you were to pick up a cup of water and shake it up, you would see that it actually is all these little individual colonies. Another thing that I hate is duckweed. Duckweed is probably one of the most annoying things that I've ever worked with. There are these little tiny plants that sit on the surface of ponds and in shallow areas of lakes. And if you've ever worn waders and gone through a pond, which I'm sure many of you have, they are just all over you all the time. It's all over your equipment. You're just covered in duckweed all the time. It can look like a harmful algal bloom because it sits on the surface of the water and it has similarities. But compare that to this, what we call like a thick soup. People call it like pea green soup, which is really appealing, I know. So, you can see this guy reaching in and he's able to actually pick up the surface scum of this. And in case this isn't obvious, don't do that. Don't do that. It's really could be bad for you. Let's see. And finally we have, oh no, but I did too. So also with harmful algal blooms, and I believe Jim mentioned the pollinating that happened a week or two ago. Wow, I am super allergic to trees and pollen in this year, just for whatever reason. I think I hit it just right. There were these crazy streaks of pollen across the lake, and I'm actually surprised I didn't hear from too many people that they thought that they were blooms, which is great. I think people are getting more educated. So it's really amazing, though, because it kind of teaches you that harmful algal blooms are dependent upon many things like water currents and wind. And so this day that I went out early in the morning, I went through what's called a Langmuir line. And Dr. Langmuir is famous for this surface chemistry interactions that he studied. And he lived right on Crown Island here at Lake George. And I took this photo a minute away from Crown Island. I was like, he would be so proud that something that he discovered and studied happened right off of his docks. So this pollen, you can compare the color of this. This color is more yellowish compared to the blue-green that is of cyanobacteria. And this photo is actually of Lake George. And you notice I say cyanobacteria and not harmful algal bloom because this is where it gets difficult, because you see compared to these previous images how, that's awful and how little we have here at Lake George. And they can be so short and ephemeral. So it tells you that we are not to this level of extreme harmful algal blooms yet. And so in some ways people are having a tough time calling them HABs. So in my opinion, I think that they are absolutely precursors to this, and there's something that we should definitely monitor and research more, because it's such a rare circumstance that you get to see the beginning of this at a lake. So more vocabulary words, and I did mention that there isn't going to be a test, right? But I will maybe grill some of you on it later. I wanted to make sure I stay on my points here. So let's see. So I want to get back down to the microscopic level, actually, because again, we were showing this on like more of a macro scale, but if you were to put these things under a microscope, you can see these different structures and why they're all important. So first we have heterocysts, and heterocysts are interesting because it actually allows cyanobacteria to fix nitrogen from the environment. This came up a few times previously. We know that plants need some basic things. They need nitrogen and phosphorus and light. And the thing about cyanobacteria that makes them so good and have these adaptations that make them so competitive is that they actually don't really need nitrogen. They can grab nitrogen from the environment. So in areas or times of low nitrogen, they can outcompete true algae very quickly. Next is an aconite. So with an aconite, it's actually a dormant cell, or more like a seed if you want to think of it that way. So after you have large blooms at lakes, what's going to happen is they're going to run out of food, they're going to run out of whatever they want, and they're going to die back. They don't live forever. But the thing about cyanobacteria is that they're able to kind of recede back into this dormant cell and sink back down to the bottom of the lake and sit there just in stasis, basically, until conditions come back, become more favorable, and then they hatch back out and regrow. They're kind of cheating. It's not very fair. Not fair to algae, I should say. And finally, we have aerotopes. And aerotopes are also very, very important and unique to this because it allows cyanobacteria to control their buoyancy. So again, all those photos that I showed you are all cyanobacteria on the surface of the lake. That isn't the only way they exist. They do live in the water column as they grow, and we'll get into that. But because they're able to control their buoyancy, they're able to float up to the surface and get as much sunlight as they want. They're able to move towards nutrients, more availability. Again, they're cheating. These adaptations are very, very good. So, and I should mention that my degree is in ecology and evolution, and so I study this kind of stuff all through college, and it's just amazing to me, and you almost have to respect it if it wasn't causing so many problems. So, what kind of problems are we talking about? Harmful algal blooms are able to produce toxins, and this is the real issue here, is that they can be unsightly, sure, but they can also be dangerous to your health. They can create antitoxins, saxitoxins, all these different toxins, and they can affect your brain and your liver and all sorts of things. So typically that's happening at really high densities, and I can tell you that there has never been a toxin present from cyanobacteria in Lake George. We partner with a chemist down at RPI, and it's great because before we used to have to send these samples off to a lab called Greenwater, and it would be, let's go get a sample, we got to ship it overnight, we got to pay a bunch of money for that. Then they don't get back to us by email very quickly, and we're bugging them because we want to tell people, is this toxic? It just created a big problem. So by getting these chemists on board down at RPI, I can go and grab a sample and pick up my phone and say, hey, Abby, what are you doing? And I just drive it down to Abby, and she runs a sample for me. It's great. So a really, really good pickup there by us being able to do that. And I included this photo here, this dog, because the reason, in my mind, that I started to hear about harmful algal blooms before I ever did research on them is because there were some dogs that had passed away at a few different lakes across the country. And that caught national attention because people were like, well, how is that possible? My dogs go in the water all the time. So it's really important to educate yourself on this and understand what to look for so that you can avoid this. And this one in particular was near Bolton Bay, actually. And so whenever the resident called me, I got there and their dog was running around, and I was like, this is a great education moment. I really think that you should keep your dog out of the water. I'm pretty certain that there won't be toxins in this, but just to be safe, I wouldn't let your dog in. And the dog was not too happy about that. So as I took all the samples that you can see lined up there, she definitely stared me down and gave me a pretty harsh review of my work. So, but even non-toxic blooms can be an issue. They can, as the cyanobacteria recedes or dies, that means that it's going to go through bacterial respiration, which eats up oxygen in the water. So this could reduce oxygen in the water. And even over at Chautauqua, as I showed you, those blooms are extremely intense on the surface, and they can last for weeks in some areas. So you're blocking all the light that would get to all the native, they're all native, but getting to all of the vegetation below the blooms. So it's really important too. And to be frank, they're unsightly and smelly. You want to come to this lake and you want to swim, you want to recreate here, you want to live here. And if you have things like that going on, it kind of ruins the whole experience. So, also important. So, are harmful algal blooms a global problem? Yes, you can literally see these happening via satellite. So I looked into this a little bit because I'm not too well educated on different countries and different continents, but it seemed like almost all continents have large lakes that are used for drinking water, and this is the problem that they're having. Harmful algal blooms are happening more and more, affecting drinking water, and that's obviously a really bad thing because there isn't normally a backup. This is where they get their water from. This one here is famous for Lake Erie. This happened in 2014 near Toledo, and they had to put a do not drink advisory out because they were saying, we're not able to control the toxins in this water, and you need to drink at your own risk. So this is when the water crisis really started to kick off as well. And I wanted to mention that there are many researchers. It isn't just us. There are many researchers around the world, and there are entire conferences focused on this kind of research. We have them here in New York because this is becoming an area of focus. So in my opinion, this must be addressed, and we must continue to do research and monitoring to understand this, because with the issues I'm about to talk about, I don't think it's going to get better. So we can get into the fun part. So how do HABs occur? And I've been talking about this a little bit throughout the different years, and I always kind of tweak it and change it a little bit, but this is because at the Jefferson Project, we're trying to understand the mechanism here at Lake George, but this isn't exactly specific to Lake George. The trick here is that we have all these advanced sensors and people so that we're able to put a little bit of our special sauce on it. So just to begin it all, as I already mentioned, external nutrients equal fertilizer. So we know that plants need nutrients. So, wow, okay. So I'm going to go through it a little bit quickly. So the important part here is now we know that Lake George is a very low nutrient lake, so they need these external nutrients to grow. The dormant cells begin growing in the early season. So as soon as ice is off the lake, which, again, that might be a problem with the summer or the winter that we just had, they're able to start getting their sunlight, and they're able to grow down on the bottom of the lake in the sediments. They live as those aconites, this right here. They live as an aconite and start to grow. And then as summer storms come through, you're going to have a lot of runoff and all these different problems that we talked about earlier because of these extreme weather events. They're carrying sediment and nutrients from lawns all throughout the lake. And in these shallow bays, you know, if you've ever stepped into a stream in the summer, it feels really cold, right? The water's cold. So because it's cold water going into a warm lake, it sinks immediately to the bottom and it runs along the bottom of the lake, basically delivering these nutrients right to these aconites so they can continue to grow. Talked about Amazon Prime. There you go. So the algal colonies, after they've grown a little bit, they begin to rise up from the bottom of the lake. You know, I mentioned those aerotopes, so that's really important. They're able to control their buoyancy, and they're saying, you know what, there's not enough sunlight down here on the bottom. I'm going to rise up a little bit. So they make their way up to the middle of the water column, and again, staying out of our sight, but still growing and growing and growing. And finally, you know, usually in the fall, you have a very calm day, and the conditions are perfect. They say, this is it, this is my day. And they all rise up to the surface of the water and accumulate on the surface. And you think about, you know, the area that they are growing in, how many millions of colonies there may be. They all get kind of concentrated in one spot by a very gentle wind. And that's usually how we catch them on the shoreline. So real quick, I'm just going to run through the differences between places like Lake George and Chautauqua, because again, I want to put this all in perspective of what's going on at other places to really kind of, you know, light the fire. So at Lake George, you can see, I was laughing to myself with a couple of these photos because I'm saying, I can barely get a photo of this. It's almost hard to see in this photo. And then I go to Chautauqua and I'm like, oh, well, that's gross. I mean, it's everywhere. It's all over the place, completely in the water column. And even our prop, you know, whenever we were starting the boat, you can see that this harmful algal bloom in the southern basin of Chautauqua went for literal miles. I went for miles of this. I was astonished the first time I saw this. So the difference really is that Chautauqua Lake is a shallow lake. It has a southern basin that is only, I think, 5 meters deep max. So there's a lot of farmland and development around it. We understand at farms they use a lot of fertilizers, there's a lot of excrement from animals, and all sorts of things that are going to get washed in during these rain events. And so their streams are delivering all this into that very shallow basin, and it's constantly mixing because there's no thermal stratification, and that's just creating a perfect mixing pot for harmful algal blooms. At Lake George, we're a little bit better. We have very, very deep lake, and it stratifies during the summer, meaning that it's kind of separated by these thermal layers. And our watershed is very small, very clean. It's mostly mountainous, right? Again, you guys can speak, you know, deeply about this. And there's a lot less development here, so we have a lot less impact. Because at the end of the day, you know, we know that Lake George, it's been here and beautiful forever. It's really going to be development that will change it. So without proper development practices, we know that it is us doing this. And so I'm just going to wrap it up really quickly with why the Jefferson Project is important to do this kind of research and why it really makes a difference and how this all comes full circle. So again, talking about stormwater runoff, because we know that runoff is affecting these harmful algal blooms. Imagine that we have a really crazy storm come through, and that is one of our weather stations on the rocks out in the lake. You can see that little spike right there. There's your precipitation event. Immediately afterwards, one of our stream stations, we can read the stream gauge that is all digital. I mean, this information is getting sent to us constantly. And we trigger these ISCO samplers so that they are taking up water samples into this shed at any given interval that we want. And again, you can see the spike in the stage height there. So that's the water coming down from the precipitation. And then with our vertical profiler, we have in the lake, at whatever depth that is, you can actually see this spike in FDOM, which is dissolved organic matter. Again, after that storm, everything running down to the stream, the dissolved organic matter is immediately picked up by our vertical profilers right here, and then you can see it taper off. So using our 3 stations, we are able to monitor every single part of a storm. Extremely cool to me. This is one of my favorite slides just to put together because it's so simple, but it really demonstrates what our sensor network is capable of doing. And we'll close it out, almost close it out with internal nutrient loading. I said I was going to come back to this plot, which now oddly has a thing, doesn't matter. There's an area of hypoxia at Caldwell Basin, and it's really the only place in Lake George that you see this. It's really strange, but basically it is a natural phenomenon. It means that it's running out of oxygen at the bottom of the lake because Again, whenever you have thermal stratification, you have a lot of mixing happening up on the top of the lake because you have all the wind-driven events. But down at the bottom, it's all separated and kind of stagnant. So just due to natural things, you're going to have all of your algae and zooplankton and stuff. As it dies, it sinks down to the bottom of the lake, gets broken down, which eats up oxygen, and that's just normal. Again, but it's normally cleared up very quickly at Lake George. It isn't really present. So for whatever reason at Caldwell Basin, this is happening. So we actually moved our vertical profiler from the site that we called T Island over on the east side of the lake from T, a little bit further north to that spot that I showed you at Caldwell, because that's exactly where this is happening in about 34 meters of water. So what we want to know is this growing? Because you can say, sure, animals and things want to avoid low oxygen water, but there's something way beyond that. If you look at some of the water chemistry here, whenever there's no oxygen in the water and it's interacting with sediments on the bottom of the lake, it actually breaks it down from, I believe it's ferric iron to ferrous. It's pouring chemistry, don't worry about it. And what it does is it breaks the bond and it releases phosphorus from those sediments up into the water that normally that wouldn't happen if there was oxygen present. So because this is really the only spot that that's happening, and that's what this plot is showing, that as you decrease in oxygen, it's going to release more SRP or dissolved phosphorus. That could be feeding harmful algal blooms. It's in the southern basin. That free phosphorus could be picked up and delivered to other parts of the lake. So really, what can you do? Let's talk about that. Because you go through this whole thing, and I know it's a lot of dense information, but you can join the Algae Watch, because we have a lot of things going on at the Jefferson Project that we're trying to understand how these blooms occur. And the reality is we don't have a lot of data to use of actual bloom events. It just doesn't happen that often. If we were at Chautauqua all the time, I'd say, sure, we can basically go out at any minute and throw a dart and we're going to hit a harmful algal bloom. But here, they're so quick, so ephemeral. So I'm really counting on all of you to talk to the LGA, talk with us, and say, hey, I think I see something, can you come sample it? And I'm probably going to be the guy that does it. So it's all good with me. And I want you to know how to identify a HAB. So again, showing you those slides earlier, I wanted to show you what is and is not a HAB. And I want to make sure you're not contributing to nutrient loading. Again, listen to the recommendations from the people before me. They know the deal. It is very important. And if you think you've spotted a HAB, make sure you reach out to the Lake George Association. With that, any questions?
We do have a few questions. The first one is, last year there was a vertical profiler at Glen Burnie. What were you guys looking for there?
At Glen Burnie up in, so I call it Blair's Bay. Blair's Bay, there's a stream there that is called Sucker Brook, and it's been one of our monitoring stations for a long time, and because of some land owner problems, we actually had to move that station. But it's a really interesting site because there, for whatever reason, further upstream, there's a lot of DOM, dissolved organic matter. So we were able to put a profiler up there so that whenever it flows into the lake, we can get a much better idea of what stream intrusion is doing. So I mentioned that with Northwest Bay this year. It was a very similar idea up there. That's a signal that we're able to pick up on, and using acoustic Dopplers, we're able to understand the water currents and the water velocity and everything, and pair all that together to get a better idea of what that stream is doing as it comes into the lake and where it's going.
All right, next question. Should we be concerned if we go swimming at our property and find some algae but not a HAB? Is it okay to swim in that?
I think it is totally fine, and if it wasn't, I probably wouldn't be here today. I swim in this lake almost every single day, and I have been in many, many bad conditions with algae, and it's just a plant. It's a plant like any other. I mean, there can certainly be some irritations and things if you're allergic to stuff, and some algae may cause irritants, but for the most part, all of this is very innocuous, and I mean, it's not exactly fun to touch because it feels like slimy cotton candy. But yeah, so it may not be the most pleasant thing in the world. And even, I mean, if we want to get real brief into this, who here noticed a lot more growth on your rocks, like things around your bay this year? Any idea why you think that's the case? It's so warm. I know, it's so warm. So I'm working on something to kind of give you guys a really fun visual of that. But we had basically no winter. There was never any ice coverage at all on this lake. That means there was no light blockage happening on the surface. So all of these plants, all these periphyton and algae and everything growing on your rocks and docks, they never got cut off from light. So they were able to grow and grow and grow, and they really extended the growing season this year, which is going to cause issues and slippery and slimy stuff.
All right, we'll do one more. Does Sunnyside Lake have this problem?
I'm not familiar. Is that the one to the northwest?
It's over in Queensbury.
Queensbury, yeah. I'm sure. So there's some basic things you could look at, like depth of the water and water residence time and those sorts of things. I'm a transplant here. I moved here about 10 years ago, but for the most part, I've focused on Lake George, so I don't know the surrounding area too well. But I would guess that it would have very similar weather conditions to us. And if there's high amounts of nutrients and it's a shallow lake, it could definitely have issues like this. And it's not spring fed. Okay. Mm-hmm. Sale. Sure. Okay, so that's interesting too. She's saying that it's a very weedy area. There's a lot of productivity. So because of that, that actually can take up nutrients that other things might need. And so in some ways, it can almost prevent harmful algal blooms and spread out the diversity of plants. And yes, Bob. Oh, okay. So there you go.
You
Yes.
can also check the CSLAP report on the DEC website and see if there's any information there, if anyone's doing the CSLAP research out there.
All good. It's the way it works. And
Thanks, Brian.
Matt, do you have a question? No.
Woo! Give it up for Brian.
Thank you all very much. Wait, real quick, I have a question. What's a sarsaparilla?
Chris, thank you so much. We're so fortunate to have the research partner in the Jefferson Project. So next, our final speaker is Tamara Van Ryn from the Adirondack Park Invasive Plant Program. Tamara?
Hello, everybody. Do you need to stand and just put your hand up for a minute? Because some of the seats, yes, okay. But don't leave your seat. No, right, the ones who are leaving their seat, just stand, put your hand up for just a second. All right, good. Actually, I'm going to talk about trees, so you can pretend you're a tree branch. There we go. All right. So, I'm going to switch gears a little bit. We've heard a lot about what's happening inside the lake, and I'm going to talk a little bit about what's happening upland from the lake and how, in some ways, that may impact the lake in the future. I'm going to tell you a little bit about Hemlock woolly adelgid, and then I'm going to talk about a coalition that we have working here in Lake George, and I'm going to leave it with some things you can do to help. I work for the Adirondack Park Invasive Plant Program, which is hosted by the Nature Conservancy. We are one of 8 regional partnerships for invasive species management across New York State. So we're very fortunate in New York State. New York puts about $14 million into invasive species management every year. And there are 8 different kinds of partnerships around the state, each one hosted by a different organization. So I am very fortunate to work with a team of really great 5 staff people, and we have 3 seasonals with us this summer as well. My background is in forestry, so I get to spend a lot of time on forest pests. We have a strategic plan, and our strategic plan calls for us to basically protect water, protect land, engage the community, and also do research and innovation, similar to some of these organizations. So we also, I'm going to talk about our forest pest work today, but we do a lot of invasive plant work on all different kinds of plants. We also have a full aquatic program, and we do a lot of education programs, and we'll be having an invasive species summit at Tannery Pond in North Creek on October 10th. So I'm going to talk a lot about invasives. Many of you already know what invasives are, but it's always good to start out with a definition. So an invasive is a plant that is not native to our region, so start with that. Okay, poison ivy, how many here really like poison ivy? No, it is a native plant, so it is not considered invasive. It's something we don't like, so we don't love every native plant, but it is not invasive. Okay, now how many people do like tomatoes? right? Okay, see, so tomatoes are not native, but we like them, and they are not considered invasive because the next criteria for it to be invasive, it needs to be non-native and then cause harm to the environment or the economy or public health. So when you think of something like zebra mussels clogging up all the intake valves, that would be an economic harm and a potential public health harm. So that's where we get to where we have things that are not native into our ecosystems, but they're actually causing harm. So that's the work that we do at the Adirondack Park Invasive Plant Program. So some people often ask, well, why does the Nature Conservancy care about this? And the Nature Conservancy is very involved in trying to protect resilient places that will be able to survive as the climate warms and create refuges for wildlife and for people. And so this is not animated today, but this just shows where migrations flow. So we're talking about migrations of birds and mammals and others, and you can see that we're really in this hot spot here of the flow of migration. So the Nature Conservancy has taken this information and mapped the most important landscapes to protect in New York State. And many of them here in the Adirondack Park are already protected, at least from development. But that doesn't mean that the biodiversity and that the resiliency is protected, because as our climate warms, this landscape is going to change. And so one of the things, because I work in forestry, looking here at the Adirondack Park, I spend a lot of my time thinking about hemlock trees. Now, how many of you can go out and identify a hemlock? Oh, quite a few. Okay, so I don't have to write. I kind of feel like hemlock is my talisman or something, right? I think I might have been a hemlock in a prior life. So hemlocks, as you know, are keeping our stream sides cool. They're also producing great cones in the winter. And this time of year is a great time to be able to tell if your hemlocks are healthy. You're going to see all these bright green tips at the end of the branchlets, and I get really excited. I've been driving around like last couple weeks, and I'm like, oh, it's tippy. Oh, they're green tips. I'm so happy. So what happens when they get fully infested with hemlock woolly adelgid is you're not going to see all those bright green tips. So I love these tippy springs that we still have. Okay, so I've talked a little bit about hemlock. Hemlock makes up about 13% of the overall Adirondack forest, but I'll show a couple slides in a minute. Here in the Lake George area, in some of our areas, it's closer to 60% of actual forest area. So it is incredibly important. And it's actually a species that, in a way, creates its own habitat system. That's considered a foundational species when you talk about forest ecology. And so what's happening is I consider hemlocks, if you will, the house that we live in these forests, right? They are the structure that keep us warm in the winter and keep deer wintering areas. You'll often see the deer bedded down there. They are the places that keep us cool in the summer. They're the places that give us shelter. They keep our soils stable. They are, again, the sort of house or the structure of our forests. I'm not going to spend time today talking about beech, but beech leaf disease is another concern. And I always liken beech to the grocery store or the pantry of our forest. So, but hemlocks are incredibly important because, as I said, they are stabilizing our steep slopes. If you're walking around here or you've seen some of these lake shores and you've got these really steep slopes, that's where the hemlocks are. And because they're keeping the areas cool in the summer, those cold water streams that we want for that trout habitat, they're cold because that hemlock is helping to shade them. And they're also habitat for an incredible number of species. And then here, Finch Prine is one of the places that actually makes specialty paper out of hemlock pulp. So hemlock has a lot of importance, and the reason it's connected to our lake is that, again, it's keeping those soils stable, it's keeping that water cool. Without hemlocks, we're going to see warmer temperatures into the lakes, potentially more erosion into the lakes, and then what happens with the chemistry of the forest is that you often see nitrogen loading coming out as those hemlocks die. So, as I said, this is sort of a map of hemlock density all over New York State, but the darkest red here in that Lake George region in southern Adirondacks is the densest hemlock. And I've had other foresters from other places of the country come, and I'm like, no, really, like, we're special here. And they're like, I've seen hemlock before. And I'm like, not like this. And we really do have unique hemlock habitat here. So there is a pest called hemlock woolly adelgid. It is not this big. This time of year, it is a teeny, 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 tiny little, barely a sesame seed, not a sesame seed, closer to a poppy seed. But when you blow it up under a microscope, you get to see this very long proboscis. And that is what it will stick into the base of a leaf needle. And then the plant feels like, oh my God, something's biting me. I'm going to wall off that wound. And so it walls itself off, kind of like you have a scab or something, but then nutrients don't flow up and down in that area of the branch anymore, and hemlock will end up dying from the branch tips down. So what it does to protect itself and is in the fall, this little poppy seedy typey thing is going to weave itself almost like a spider might, but weave itself a little mass of wool, and that's where it'll lay its eggs, and it keeps it cozy warm in the winter. It doesn't like super cold temperatures. We don't get those cold temperatures anymore. So you can often identify it because these masses, these woolly masses, will persist year in and year out, and you'll see, even though they're not active right now, you can see evidence of last winter's. We do ask folks to not do a lot of surveying right now because there's only one time a year that this little seed bug stage of the insect is mobile, and it's usually mobile from about April to July. So we hold off and don't do surveys until later in the year. Okay, so hemlock woolly adelgid, though, it came over most likely on a plant from Japan. There are Japanese hemlocks. There's hemlocks in other parts of the world. There are adelgids in other parts of the world. There's adelgids for the hemlock in the West Coast, and those trees survive just fine. But our trees here, our eastern hemlock, are not resistant to this Japanese adelgid. So it likely came in the south, and it spread its way all the way across. So this is hemlock range that's not infected. This is hemlock range that is infected. So, and in New York State, this is just areas that have infestation, and here we are now in the Lake George watershed, which I'll talk about in just a minute. Just to give you a bit of a picture, though, this is Great Smoky Mountains National Park, and they have now had hemlock woolly adelgid for quite a while, but I had the, I don't know, misfortune maybe of hiking there maybe close to a decade ago now in the spring. And I had never seen hemlock woolly adelgid. I'd heard about it, right? I'm hiking, and I was in this forest, and I'm like, it is just ghostly. It's completely springtime, and everything should be, you should be hearing birds, you should be seeing all kinds of things, and all of this is dead hemlock. This is not hardwoods. These are dead hemlocks. So at Great Smoky Mountains National Park, they have lost most of their hemlocks, only the hemlocks that they've chosen to save by doing both a combination of chemical and biological control treatments. So what's on deck for us here? The thing is, as the climate warms, those bugs will be able to survive longer. So we go out and we actually collect those little woolly samples and tease them apart under a microscope and count how many of those little adelgids are alive every spring. So this year, we were only seeing about 20% mortality. 20% of those little masses had dead bugs in them. The rest were alive. We'd love to see 90% mortality. They don't like it when it's below like 25 below, but we don't get below 25 below very often anymore. So as you see that the climate warms, this is 2015, these are projections, 2015, 2030, 2050, we will see hemlock woolly adelgid surviving everywhere. And here you can see the dark brown is where hemlock is thriving. And so you can see we have a lot of dark brown. You can still see here we have a lot of dark brown. By the time we get to 2030, the projections are we may only have hemlock in sort of the coldest hollows in the Adirondacks. which again has a lot of impacts for our water bodies because again, you can't separate the lake shore from the lake. So I don't want to be all gloom and doom though, because I fully believe that somewhere in these woods is the key to survival. There have to be resistant hemlocks. We're going to find them, but we have to keep enough hemlocks alive long enough for us to find that key. So this, is it going to animate? No, it's not going to animate. Okay, so this is just talking about, again, I've sort of talked about what the different impacts are for hemlock in Lake George. The one thing I didn't mention was also two things, I guess, which is hazard trees along the road. In Great Smoky Mountain National Park, they had to close off one of their park roads for 6 or 7 years because they had so many hazard trees, they couldn't get them all out of the road. And so when I talk with folks like highway departments, I say, you know, this is going to affect your municipalities as well. And then many of you remember the spongy moth, what now, 2 or 3 years ago. And we got so many calls, you know, the hills are dead. Why does it look like this? Well, I think as we see the hemlocks begin to die, people are going to have some of the same visual concerns. So back in 2017, there was one tree noticed that was infected on top of Prospect Mountain. Great, people went out. There was a chemical treatment of about 7 acres, thought that was stopped. Unclear whether it was stopped or not. But in 2020, there was a camper on Glen Island Campground who who saw it, noticed it was hemlock woolly adelgid, and put a point in IMAP invasives. Anybody here use IMAP invasives? All right, a couple of hands in the back. It's pretty simple. It's IMAP invasives. You can just Google it. It's a very simple thing you can have on your phone. And if you see an invasive species, you take a little picture of it and send it, and you fill out like a very short little form, and it goes into a statewide database. I love IMAP. I use it all the time. And then I get alerts when when they find a new invasive. So yesterday, I did have a call from a landowner in Bolton Landing. I did go over and confirm it was hemlock woolly adelgid. I was able to put it in IMAP invasives, and all of my colleagues at DEC and at the Hemlock Initiative, real time, everybody knows and can see it's there. So we do, again, aquatic work, other terrestrial work. We found out about the first infestation of stiltgrass in the Lake George watershed from a landowner who knew what stiltgrass was and put it in IMAP invasives. This could have gone unnoticed for a whole lot longer if we didn't have somebody who knew IMAP invasives. So future training option is IMAP invasives, but it's real simple to use, and there's some information on our website, and I have some information that I can have at the table later. But what we know now is that there was a big infestation here, done continued surveys. It's now jumped the lake, so it is on this side of the lake. It's on the tongue. It's on the lake. Clark's Hollow Bay is furthest north as far as we know right now. So again, that map is not showing, but you can look at it real time in IMAP invasives because you can screen on it and check the maps. Oops, nope, that's not going to go. So there has been a lot going on with hemlock for the last 4 years. We've done a lot of on-the-ground monitoring with DEC and others and our volunteers. We've surveyed close to 4,000 areas. We've only found positives in a few hundred of those, but they are definitely concerning. And then with help from DEC and the Nature Conservancy and Lake George Land Conservancy, about 10,000 or so trees have been individually treated with a chemical treatment. The chemical treatment will keep them alive for about 7 years, we hope. Nature Conservancy owns Dome Island, so we have treated about 1,700 trees on Dome Island. It's pretty heavily hemlocked, so we hope those trees will stay alive, because if you ever boat around Dome Island, you know that's what you're mostly looking at is those hemlock trees. So there's other really encouraging stuff, though. Like I said, I don't want to be all doom and gloom, because there are some hope. There is hope on the horizon. And again, we got to keep enough hemlock alive long enough for us to find these solutions. So there are 2 types of insects that are very carefully controlled by the U.S. Department of Agriculture that have been approved for release, and they actually eat the adelgid or the eggs of the adelgid. One of them is a Laricobius beetle, and another is a silverfly. So the Laricobius beetles About 4,000 of them have been released, and now close to 9,000 silver flies have been released. So in various parts around the lake. And in other parts of the state, those Laricobius beetles have now begun to sort of self-produce and spread. So they were released at Minnewaska State Park a number of years ago, and now they're found throughout the Shwangunks in other areas of hemlocks. They've created enough populations that they can spread. So that's the long-term hope, but it is a long game. And again, we have to keep enough hemlocks healthy in order for that to happen. So a lot of other projects have been going on with funding from the Fund for Lake George. We helped do a remote sensing project. Could we figure out whether you could actually use remote sensing, aerial imagery, satellite imagery to find this hemlock woolly adelgid before anything else did? Great technology, not there yet. Found lots of dead hemlocks, but wasn't able to pinpoint that it was hemlock woolly adelgid. It was beavers. It was something, it was a logging job. It was many other things. We've also been trying to study the DNA of hemlock and collecting branch samples with Cornell University to see if we can find it that way. Again, the technology's going to come, but it's not here yet. So the best way to find hemlock woolly adelgid is to go out in the woods and look for it. And we fortunately have a lot of people right now doing that. So part of what we have came to the conclusion of, though, about last fall was there is no way that every single tree that has hemlock woolly adelgid in the Lake George watershed is going to possibly be treated chemically, and the biological controls are not going to be here quite fast enough. And so if we think that we're just going to continue, okay, DEC's got a week, they can do a treatment there, or, the Nature Conservancy can go out and inject, 50 trees this fall, scattered all over, that's not a unified approach. So working with Lake George Association, with Warren County Soil and Water, Hemlock Initiative at Cornell, very strong partners with DEC and us at the Nature Conservancy, Lake George Land Conservancy, there's a whole bunch of us in there. We've said, well, how could we be more strategic? Can we figure out a system to think about what is it that's most important to protect the lake and the communities along the lake, and figure out maybe we can come up with priority hemlock areas where we will focus our monitoring and our treatment. So we've been working on this priority hemlock area strategy, and again, all of the folks on this coalition. So, oops, I keep doing that. You can't read any of this, don't worry about it, but I'll talk you through a couple of things. So what we did is we did a lot of computer-based mapping, but we did that based on criteria that this group of people all put together, and then we ranked them. But it's things like important view sheds. It's things like the 5 most important watersheds and keeping those streams cool. It's things like the drinking water supplies, like up at Edgecombe Pond, and a number of different factors like that, old growth forest habitat. And all of those were layered together to come up with some priority scores. So each sort of pixel in the watershed is now ranked, and we'll be using these to select areas that will become the focus of 2024 monitoring. And we're really fortunate that the state, DEC, freed up money to hire a 2-person conservation crew through Cornell. So we've got 2 more folks coming to this area. They'll be living in they'll be working out of Warrensburg. This was like a huge shift in this project last fall. And the Lake Champlain, Lake George Regional Planning Board has some funding coming through Lake Champlain Basin Program to help hire a community educator and to do some additional mapping. So pieces are coming into place. There's no grand plan. There's no $1 million dropping on this yet. But, I'm tutoring a Spanish student, so we talk a lot about poco a poco, little bit by little bit, right? We're getting there. So we expect that, again, with our staff and DEC staff and volunteers from LGA and volunteers from the Nature Conservancy and folks from Lake George Land Conservancy, we've got about 125 areas we want to be able to get out in the survey this summer and understand what's happening with them. And we think through this new coordinated effort, we're going to be able to get to those. And then when it gets to fall treatment season, we're going to be, again, be able to accumulate all those resources and put it into the most priority places instead of every agency going out and doing their thing. So we're really excited about that. Today, I'm going to give you some ways that you can help. We are also, Soil and Water Conservation District is having a program here for landowners and foresters and others on September 21st, probably right in this same room. So if you want to learn more about it, that's another place. So one of the things we do is we have volunteer forest pest hunters. Our volunteer forest pest hunters, this year they adopted over 30 trails and entered almost about 1,000 points into IMAP. Of that, we had a little over 100 of new sightings that are really important for us because that's how we know where it is, how far it's spread, where do we need to go next. And it's great work. We ask people to do that again. You can start doing it even as early as, I would say, safely late July, August, and usually our forest pest hunters go through the end of March. But especially if you like to be out in the winter. All you gotta do, it's a pretty simple protocol. I'll talk with you more about it. You walk for 10 minutes, you stop, you find a hemlock tree, you find a bunch of hemlock trees, you turn over some branches, you shine bright lights up in the trees, you look for 10 minutes, walk for 10, survey for 10, enter your points into IMAP. Pretty simple, but incredibly important. So if you want to become a volunteer forest pest hunter, I can give you more information, and we'll be doing a training for our beech leaf forest pest hunters come August as well, and they'll be out in the woods this fall too. I'm not going to talk too much about IMAP, but since you are not all familiar with it, you can go to www.imapinvasives.org. You can click on New York. You can set it up on your desktop computer. It's easier to set yourself up an account on your desktop computer, but then you just put it on your mobile app. And I am not the biggest technology person, I will admit, but IMAP on my phone actually works. So you get your account set up on your desktop, then like yesterday, I just go here, I'm gonna record a new species, gives me a couple of things to click, I record it. I have no service because I'm someplace like Chinchacook, doesn't matter, my phone remembers it. When I next get to a place where I can upload, I upload it, and as I said, especially if it's a new find or an important species like this, a whole bunch of people get notices. So, first thing you would do is log in, and again, I have some instructions for this, so I won't spend a lot of time on an IMAP training here. But really, if you have any interest in just looking for anything, you can put your milfoil in there. You can put in Asian clams. You can put in zebra mussels. You can put in knotweed. You can put in wild parsnip. All of that data is incredibly helpful for folks who are managing. So sign up on your desktop. You can, if you want to do forest pest hunting, you can sign up for a project for the APIP Volunteer Forest Pest Hunters. A lot of folks can help you with this. If you're interested, I can help you today and give you some more information. So again, then you just take your observations. So I think collectively, we have an incredible coalition in Lake George that doesn't exist really anywhere in the country. We've been able to get ahead of a lot of other places by working on this priority hemlock strategy and being able to pool resources. We have a lot of concerned citizens out there who can be our eyes and ears on the ground. And we got Laricobius beetles and silver flies that are hopefully finding tons of food out there right now and are going to be the hope for our future. So that's a little bit about hemlock woolly adelgid, and I'll be around after the program. I'll take any questions.
We have a couple questions. First, how are hemlocks chemically treated?
Hemlocks are chemically treated with a combination of two chemicals, and generally they're treated with a basal bark spray unless they're very close to the water, in which case they are injected. The injection is a lot more time consuming and expensive, but the basal bark spray is literally a backpack sprayer and a wand, and you spray the bottom 4 feet of the tree and you walk around it, and it goes directly on the bark, taken up directly by the bark. So it's not high aerial spraying like you might see for some other kind, like you would see if you wanted to treat for spongy moth.
Great. Next question. What should homeowners do if HWA is on their property?
Yep. So the good news about hemlock woolly adelgid, different from emerald ash borer, is that hemlock woolly adelgid is probably not going to kill your tree immediately. It might be 5 to 10 years. So one of the things you can do, you can hire a private pesticide applicator to come treat your tree. There are many of them in the area. and some of them can do injection, and some of them, if they're close to the lake, or basal spark spray. So you can save your individual trees. We do not encourage you to go cut all of your hemlocks. Please don't do that, because again, it is a slow progressing pest, and so if you have a commercial timber stand, you can definitely thin that down and take some of that hemlock out, but you don't want to create too much light, because here you're likely to get barberry and hay-scented fern and a bunch of other things coming up if you give it too much light. So as a homeowner, you want to keep your eye on your tree. If you want to save it, right now your choice is to hire a private pesticide applicator to treat that tree. But you should also put a point into IMAP invasives, because that helps us know how far it's spread.
Great. Last question. Can a replacement tree be planted to take the place of the hemlocks?
That is a great question. So there's nothing quite like a hemlock that is going to provide that kind of structure that that hemlock provides. But if you're an individual homeowner and you can keep the deer out, which I know is its own issue, if you were to plant some of the cedars along the lakeshore, you would get that same sort of cooling effect and that kind of year-round evergreen effect. The other thing, that if you're planting more for trees in your yard, in your landscape kind of thing, red maple is certainly, red and sugar maple are trees that are not currently infested by things, and that's what we're going to see most naturally come up. In our forests, I expect we're going to see a shift to more maples and birches as the hemlock and the beech drop out. But again, some of those conifers, people have also had some success with some red spruce. That kind of just depends on whether you're at the right elevation for that to grow.
Thank you so much.
All right, and I do want to do a shout out to Monica and Lindsay, right? Because this slideshow for all 5 presenters was incredibly professional and so smooth, and I know that what I sent them did not look so good. And so they made us all look good today. So a special shout out to Monica and Lindsay.
Thank you, Tamara, and thank you, LGA staff, and thank you, Camp Chingachgook. And so we're going to hear, I know I'm trying to pronounce that. Sometimes I hit it, sometimes I don't. So Tiara McGuffey from LGA is going to talk about other programs that we have going on this summer, and then we'll be around for about a half an hour to answer your questions individually around the building. And thank you again for participating.
Sorry. Thank you, everyone. I'm Tara McGuffey. I'm the Development Director for the LGA. And I want to just quickly thank all the presenters today. I think, how many people in this room are members of the LGA? If you're not, we would love you to become a member of the LGA, learn more about what we're doing, and help in any way that you can. However, we have a science to solutions model, the LGA does, and you saw every single aspect of it here today. Obviously, we had a lot of education. Your minds are probably blown in many ways right now. Innovation, you heard about all the innovation. Investment, you heard about all of the programs we've invested in. But most importantly, what I took away from today was the incredible importance of our partnerships. Our partnerships with municipalities, our partnerships with other organizations, our partnerships with businesses, our partnerships with you as homeowners. Whether our motto at LGA is we protect Lake George together. We can't do it alone. And all of these other organizations are also protecting Lake George. They're protecting what we love. This beautiful lake, everything that's in our heart and in our souls. So I will talk about I just had to say that, kind of finish that up. But I will talk about some of the events and things that we have going on. The 4th Wave Summer Kick is tomorrow night. So join us at Lighthouse and aboard the Adirondack if you want to take a cruise with us. It's going to be fun. It's going to be a lot of music, lots of nibbles, and just a good, fun night. We have wave training. You can read them all here. We have some open weekends at the Bolton Historical Museum. Floating classroom public programs. If you haven't been on the floating classroom and you want to go, take your kids, your grandkids. It's so educational, so fun. And then you can see all the different things that you can be a part of. So whether you're volunteering for us, whether you're volunteering for the Nature Conservancy, the Land Conservancy, we're all here to protect Lake George. Salt Summit we talked about. Okay. And then after this, we're going to have meet and greet. And Stormwater, Jim will be here. Oh, and by the way, Tamara and Jim and Phil will be in the other room, and our staff will be around this room. So if you want to talk to Chris about the water quality, septic programs, land use, if you want to talk to Monica about any of our over 40 programs that we do, if you want to talk to Lindsay about education, Danica and I will be sitting here selling swag, and it's very cool, by the way. And also, you know, if you have any questions about development or funding, you can talk to us about that. Tamara, the Willie Adelgid, and then also Brian for the Jefferson Project will be around. So thank you so much for coming. Thank you, Camp Chinjacook. My children went here. We love the program. We love this place. And by the way, Janae, right? Is that? Lindsay and Monica have been just so happy with everything and everything you've done for us the last 2 months to make this happen. We really appreciate it. Thank you to Camp Chinjacook for having us. So.
Also, the staff of Camp Chinjacook have very generously offered to give anybody tours of the camp if they would like. Also, shout out to Black Mountain Visuals. All of this information in the presentations will be on the YouTube if anybody wants to go back and relook, or I know it's a lot of information that was presented to you guys today, so it will all be available on the YouTube channel. Thank you guys.